There have been many studies that have looked the effect of genistein, the isoflavone in soya, on the breast and trying to understand whether genistein has estrogenic effect, harmful effect on the breast. And I list here 4 different studies that have looked this, and 2 of them have observed estrogenic effects and 2 have observed no estrogenic effects. And I want to briefly go through these 4 studies. So in the first study, that was a 6-month study where women were given 38 grams of soy protein isolate daily. That contained about 38 milligrams of genistein. And it was shown to increase the volume of nipple aspirate fluid. And the fluid also contained hypoplastic epithelial cells that are precursor cells for cancer. And since the more NAF volume a breast generates, the higher the woman's risk of getting breast cancer, this study suggested that there might be some adverse effects on women. The second study was only a 2-week exposure. In that study, women were given 60 grams of soya protein daily, and that contained about 45 milligrams of isoflavone, and it was shown to increase the expression of PS2 in the nipple aspirate fluid, and PS2 is a standard gene that is looked for estrogenic effects. When it increased, that means there's an estrogenic activity. Then the studies that found no estrogenic effects, in the first one, women who had biopsy were given isoflavone tablets that contain 200 milligrams of isoflavones between the biopsy and then when they had the surgery. And the endpoint in this study was to look the ratio between cell death and whether the cells were proliferating, and there was no change in that ratio compared to some historical control samples. And that was thought that this indicates that there is no estrogenic effect. However, there can be an estrogenic effect even though this ratio is not altered. The last study is my favorite study. That is a 6-month consumption study where women were giving 2 servings of Asian soya food products, which generated 50 milligram isoflavone concentrations. They obtained 50 milligrams of isoflavones per day, and this mimics very well the typical Asian diet. And this 6-month exposure didn't have any effect on the volume of a nipple aspirate fluid or estrogen levels in the nipple aspirate fluid. So you can see that there are different results, and it's very difficult to make any kind of conclusion from these studies, perhaps mainly because most of the studies have used isoflavone supplements rather than feeding women soya foods. And perhaps instead of focusing only on the estrogen receptor and direct estrogenic effects, we should look beyond the estrogen receptor and what happens after it's activated by genistein. It has been found, for example, that genistein inhibits activity of on the top of your screen you see NF-kappa B. That is a gene that induces inflammatory responses, and it is very important in cancer, and when it is inhibited, that is a good thing. That improves the immune system and prevents cancer. Genistein also upregulates many tumor suppressor genes. In cancer, we think that it is a result of tumor suppressor genes being downregulated and oncogenes being upregulated. So with genistein, we see upregulation of the tumor suppressor genes, which makes the normal cells, or perhaps the cells that have already undergone a little bit of changes, to fight better for being turned to malignant cells. And one of these tumor suppressors is BRCA1. It is a gene that is linked to about 30% of familial breast cancers when it's mutated. And when it is activated by genistein, it can lead to upregulation of another gene that is called DNMT1, that is a DNA methyltransferase that is important in regulating epigenetic machinery. And when it is upregulated, it means that some of the genes can be silenced by increased methylation. And the genes that can be silenced could include these oncogenes, and those are now on the right side of the screen. For example, HER2 is one oncogene that is closely linked to breast cancer, and genistein downregulates HER2. So genistein and soya can have multiple different effects, and these effects can explain some of its protective abilities. Now, if we look at the studies that have been done looking soya intake and breast cancer risk, there are many of them, and there also have been 3 meta-analyses that have combined, looked all the individual studies, and these 3 meta-analyses have made very similar conclusion that soya intake is associated with 15 to 25% significant reduction in breast cancer risk among Asian women, and particularly among Asian Americans. These studies also show that soya intake in Caucasian women hasn't been linked to reduced breast cancer risk. And this might be because soya intake in Caucasian women is lower than it is in Asian women, and many Caucasian women start consuming soya, for example, first time close to the menopause, trying to deal with their menopausal symptoms. So why the intake in Asian countries might reduce, or Asian women might reduce breast cancer risk? It is possible, or the results suggest that the intake around puberty, before puberty, and shortly after puberty of soya foods is the period when most of the protection is initiated against breast cancer. There are several animal studies, preclinical models that have been done that are very consistent in showing that in these models prepubertal exposure provides lifelong protection against breast cancer. There also are 5 studies that have been done in humans that show that if a woman was consuming high levels of soy during childhood, she later on had significantly reduced breast cancer risk, and those women who consumed soya also during adulthood had the lowest. So if a woman who consumes soya throughout their lifetime, starting in childhood, seemed to have significantly reduced risk of developing breast cancer. And why is that? We know that the soya intake in childhood might reduce the aggressiveness of the breast tumors. So even though the exposure occurred during childhood, it permanently alters the programming of the mammary gland, so that when the tumor develops, that tumor has a lower grade than tumors in women who haven't been consuming genistein, or in this case animal models, and the mitotic index is also lower. In addition, the critical hormone receptors, estrogen receptor, progesterone receptor and HER2 are all expressed at a low level in the tumors in preclinical models if the exposure to soya foods or genistein occurred during childhood. And there is also one human study that has looked childhood soya exposure and changes in the tumor breast cancer characteristics, and they also found that HER2 expression was permanently reduced in the tumors. There also are studies that show that that have caused some concern about the safety of soya and genistein for women who have been diagnosed with breast cancer. So the study is done by Dr. Helfrich. First, he did a cell culture study where he used MCF7 estrogen receptor positive human breast cancer cells. In a culture model, he treated them with estradiol or increasing doses of genistein. And the doses of 1 micromolar of genistein, that is representing the Asian soya food intake, what individuals who consume soya at the Asian soya food consumption level have in their blood, that stimulated the growth of those breast cancer cells. She then transplanted these human breast cancer cells to a nude mouse model and ovarectomized the mice and then treated them with estradiol or fed them genistein in the diet, and after a short delay the genistein started stimulating the growth of the human breast cancer cells.